Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. For those who don't know me, my name is Aufa Humaira with my last ID number 040 and I'm from Biology Education B. Today I'm going to be talking about Xanthoprotein test and how does it work. So, what is Xanthoprotein? Xanthoprotein is qualitative test on the protein used to show the presence of the benzene group. And what is benzene? We can say that benzene is a cyclic compound formed of 6 carbon atom bond in a ring. This compound is nonpolar, colorless, flammable, and insoluble in water, but soluble in sulfur. Next is the purpose of Xanthoprotein. The purpose of Xanthoprotein test is to determine the presence of tyrosine, phenylalanine, and tryptophan in a protein. Let's move on and discuss about methods. In this experiment, using qualitative methods and analysis, data collection techniques by conducting direct experiments and through laboratory assistant explanation. So before we start the experiment, we must prepare some tools, materials, and reagents. So here's the materials and reagents from these experiments. The first is we must have 2% albumin solution and 2% gelatin solution, 2% casein solution, and 2% of phenol. The reagent that we use in these experiments are nitric acid and sodium hydroxide. Some tools that we use in these experiments, namely test tubes, measuring cups, pipette, spirit lamp, and test tube clamp. So here's the steps of this experiment of Xanthoprotein test. The first step, we must prepare for test tubes in clean and dry condition. Then fill each tube with 2 ml of the material to be tested each. After that, add to each tube containing the solution test with 1 ml of nitric acid reagent. Then we observe for a moment what happened. After that, heat each test tube directly using the test tube clamp over the spirit lamp. Warning, do not let it boil. After the solution starts to change color, clean the solution tube with water. When it is not hot, add approximately 5 drops of sodium hydroxide slowly to each test tube. Then, what will happen? So here's the result of Xanthoprotein test. At this point, I'd like to turn to discussion. So, nitric acid solution which function to break down protein into benzene groups. Amino acids that show a positive reaction for this test, namely tyrosine, phenylalanine, and tryptophan. Protein containing amino acid residues with phenyl radicals in their chemical structure when added with concentrated nitric acid will form wet clumps. Orange color appears. On heating, the color of the wet clump will change to yellow, which eventually turns orange when added to an alkaline solution. This is where Aufa or sodium hydroxide works, because with the addition of Aufa, which function to make the environment alkaline, so that an orange color can be formed. Let's move on and discuss about why isn't the reaction working. So in the first reaction, during the experiment, there was a measurement error. Second, when the test tube is heated for too long, the reaction does not work optimally, and the reaction may even be hampered and damaged. Let's review the key points from this session. First, albumin, gelatin, and casein can produce a yellow-orange color because they contain benzene nuclei, namely tyrosine, tryptophan, and phenylalanine. Second, the peptone, although not a protein, contains a benzene nucleus, so it can produce a yellow color. Now we come to the end of this session. I apologize if there are still many mistakes, and thank you for your attention, thank you for your listening. May all those perhaps shall be beneficial for all of us. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.